So each amino acid has its own characteristics according to the R group. However, some amino acids are difficult to place in a particular group, and you may see different assignments in different textbooks. For example, histidine, which is weakly basic, but tends to be uncharged at the physiological pH. For example, in your body, it tends to be not charged, but it's weakly basic, oddly enough. Cystine's polarity contributed to the sulfur-hydryl group, is modest, and it is quite weak acid and can form weak hydrogen bonds with oxygen and nitrogen. Glycine is another example. Because of its R group is hydrogen, that makes no contribution to hydrophobic interactions. So oddly enough, but we're going to keep it very general, very traditional, very demure. And we're going to follow one textbook, which is, don't remember which was the name, but I'm going to list that. Okay, let's start with nonpolar. One. Nope, let's nonpolar. And nonpolar, first of all, they are hydrophobic. Remember as water fearing, right? They're like, when you're like, oh, I have phobia of heights or spiders. That's how you should remember the hydrophobic. It's like it's scared, like it has phobia of hydration. Like it doesn't want to be there, like for water. And hydrophobic amino acids, they're usually, found in protein interior. And to a question as to why, because protein needs to move around the cell, and like how would it move around the cell that's mostly aqueous, which has like around 80% of water? How would it move if its molecules are scared of water? Like they can't interact with water, so they're just going to be stuck and polymerized, and wouldn't let the protein to function. So protein would actually hide its hydrophobic ends, hydrophobic amino acids. And they're also called as hydrophobic pockets because the protein hides it. It doesn't want to expose it because otherwise it's not going to, move and work and function. So another feature is they do not accept or donate protons. So think of them as that cheap ass aunt or neighbor or friend that you have who doesn't like to spend much money. That's the group you want to go for. They don't like to donate or accept any proteins. They're very diva, like they can handle it themselves. They don't need anyone's help, and they're not going to help anyone. Like that's how they are. Um, another thing is they actually do not participate in hydrogen bonding and ionic bonds. Okay, so that's like very nice to memorize. Like once you learn which, amino acids don't do this, like if you're gonna have a question and one of these amino acids will be part of it, you're like, I know these guys, they do not form hydrogen bonds or ionic bonds because they're that person that who's cheap and doesn't like to do much. Uh, I don't know. That's very stupid for me to say, but that's how I would memorize. Like make up a story. Think of them as living things. Okay, alanine, valine, leucine, isoleucine. They tend to cluster, stabilizing proteins. Through hydrophobic interactions. So that's like another thing that hydrophobic pockets have. They interact with each other. Um, and it's actually considered one of the molecular forces. Quite weak, but it's there and it helps to keep protein, you know, stay together. Um, Another is the proline. The proline R group has a cyclic structure. And we're gonna write them out and try to figure out what's you know what are they and how do they look. Conformation. And interesting enough, in contrast, because glycine has hydrogen as its R group, allows protein flexibility. Okay. Oof. Okay. Let's write out the structural formulas of theirs. What? What is that? Why are you not? All right. And let me write here. Part one. No, it's ugly. Okay. Oops. Part one. Okay. So let's start with non-polar side chains. Let's start with glycine. It's known as gly or just G in its alphabet, and it's just nitrogen, carbon, oxygen, carbon, hydrogen, oxygen, bond, And remember this one. This is our peptide bond. Okay, no. But this is the peptide bond. Yeah, it's here. Yeah, try to like remember each one of it. And this is where, you know, you're gonna have your, this is your C terminus, and this is your N terminus. Right? You got it. Like, this is where, like, you're gonna have it. All right. This is gonna, what this was one. Let's do two. Alanine. A. So it's nitrogen, carbon, carbon, oxygen, hydrogen, hydrogen, and here it's CH3. Again, geez, our, wait, okay, nice. And our peptide bond, like, be very peculiar about it. Like, try to like write it out in every single time you see it, like valine, val, or just V. So remember again, valine, alanine, leucine, and isoleucine, they tend to cluster. Why? Because they're hydrophobic. Huh? See? Good. So, and okay, this, let's do it again. Okay, no. Like, you got it. This is peptide. They don't form, again, they don't form hydrogen bonds. Why? Because they're hydrophobic. And why do they not form hydrogen bonds? Right? It was hydrogen. Right. Hydrogen and ionic bonds. Because they do not participate in donation and acceptance of proton, just protons, not proton ions. Jesus Christ. I'm just so tired. You know what? Okay, this is the hydrogen, another hydrogen. We know again, this guy is our peptide bond. Then we go for the CH3, CH3. This is our guy who's like, the R group. Okay. Not 6, it's 5. The second would be isoleucine. And he's basically, it's the same tail, oxygen, hydrogen, hydrogen. And then we're going for like CH2, CH3, CH3. Again, where is our peptide bond? It's right here. Okay, now number 6. It's proline. And proline is like quite common amino acid, like in textbooks and exams. So highly recommend you to look closely into it. Oh, sheesh. So proline doesn't have hydrogen, like second hydrogen, like it always would. Okay. Uh, it actually does have like peptide bond here. 7 is phenylalanine. A or just F. And C. And it's just like this benzene ring. Okay, you got what I mean. Okay, then it's tryptophan and methionine. And this is our peptide bond. Again, just like repeat, have an repeat. They're hydrophobic. They don't form hydrogen bonds. They don't form ionic bonds. They do not accept or donate hydrogen, you know, not hydrogen protein. It is kind of like hydrogen ion. It's a proton. Okay, 9. And they only have 9 amino acids. Methionine. So to repeat, summary. One, hydrophobic. Two, don't accept or donate H plus protons. Three, don't form hydrogen or ionic bonds. Four, there are only 9 amino acids, and they are glycine, alanine, leucine, iso, isoleucine, proline, phenylalanine, tryptophan, and methionine. That's it. That's like all the guys we have. Okay.